It is a pleasure to be here, albeit virtually and pre-recorded. I hope for your sake that my talk is either so gripping that you scarcely notice I'm not in the room, or else so effectively soporific that you wake in 20 minutes feeling well rested and ready to engage in the poster session. I expect, however, that the outcome will be something intermediate, and I owe you my sincere thanks for your patience and attention. I also thank the organizers of the conference for the invitation to present today. And the work I will be presenting was energized by thoughtful collaborators and funded by the USDA NIFA and EU Project Safeguard. Flowers are food, pollinators are florivores, and what we are doing here is trophic ecology. My title might strike some of you as provocative. For others, it may seem merely to state the obvious. For me, though, it is first and foremost autobiographical, and the story it encapsulates is the content of this talk. The story begins in the richly variegated urban environment of Philadelphia, Pennsylvania, where I used honeybee colonies to sample the composition and dynamics of floral resources in a context where traditional methods of floral surveying simply don't work. From there, I'll take you to Germany, where I nominally studied bumblebees in the Alps and actually sat at home crunching data in a home office that was really just a home. I'm the one under the sheet in the background there. In between bouts of data analysis and rather less successful bouts of language acquisition, I spent time reflecting on what I had learned about floral resources in Philadelphia. In particular, I became interested in the implications of these findings for the emerging specter of floral resource competition between pollinator species, and especially between honeybees and wild pollinators. Since I couldn't do fieldwork, I wrote a synthesis paper, and I'm going to give you the Cliff Notes version of that. In July of this year, I finally returned to the field, this time to a set of protected grassland remnants in central Germany, to examine empirically one of the questions raised in my synthesis paper, namely, are floral resources substantially depleted by the insects that visit them? Finally, I will come full circle and try to persuade you that what I found in the grasslands of Germany can guide our interpretation of the floral resource patterns I saw in Philadelphia. So, we begin with Chapter 1, Philadelphia. In my doctoral work in Ohio, I tried to understand honeybee biology from a landscape perspective. When I came to Philadelphia, my goal was essentially the opposite, to understand a landscape from the perspective of honeybees. Urban landscapes have always fascinated me with their complexity, with their seemingly infinite fractal-like depth of variation and pattern. Now, it's all well and good to be fascinated by such complexity, but it's quite another thing to go out and attempt to sample it. And if you do, you'll find out very quickly that transects and quadrats will, for so many reasons, not get you very far in a dense urban environment. Honeybees, with their ability to fly and trespass with impunity, get you considerably farther. So, I partnered with Don Shump of the Philadelphia Bee Company to set up an array of research apiaries throughout the city. At each apiary, we equipped 3 colonies with data logging hive scales and pollen traps. By analyzing colony weight dynamics and identifying the contents of monthly pollen samples, we inferred the net volume and botanical composition of floral resources in Philadelphia as seen through the lens of honeybee foraging. The most salient finding that emerged was a pronounced pattern of nectar flows and dearths, each corresponding to a phenological phase of the floral community. In the graph, the curve depicts weight change through time, so positive values represent periods of weight gain, and negative values periods of weight loss. The general pattern is that we have 2 strong nectar flows in the spring, followed by a leveling off of weight change in July, and then a strong dearth period in August, followed by a brief flow in September, and after that, that's essentially the end of the foraging season. Each of these weight features is, of course, associated with a certain suite of floral taxa. And significantly, these suites of floral taxa vary not only through time, but also through space across the different Philadelphia neighborhoods I sampled. Now, there is a lot I would love to say about this project, but I need to hasten along to Germany. I encourage you, though, to read the paper if you're interested. The important thing to know is that I became rather obsessed with these colony weight curves and the dimensions of spatial and botanical variation that lay behind them. Floral resources through time, through space, distributed across botanical species and functional traits. These patterns were complex, to be sure, but they were also tractable, and I became increasingly convinced that this multidimensional topography, if you will, of floral resources lay at the very heart of plant-pollinator ecology. This is my back-of-the-napkin version of the conclusion that I drew from this study, the simultaneous variation of floral resources through space, through time, and then distributed across taxonomic and functional dimensions. Now, at this point, you may be thinking that all this is fairly obvious, and perhaps it is. Yet, as I combed through the literature, I discovered that an explicitly trophic perspective on the relationship between flowers and pollinators was strangely elusive. There is a rich tradition, of course, of what can be regarded as pollination ecology sensu stricto, studies concerned specifically with pollen transfer and its consequences for plant reproduction. In such studies, the fact that pollinators visit flowers for food is merely incidental. The important thing is that they move pollen around. Then there are a great many studies concerned with the structure and logic of plant-pollinator interactions. In these studies, flowers and pollinators are understood chiefly as nodes connected in a graph, and the trophic significance of their interactions is at best an afterthought. And of course, landscape perspectives on pollinator ecology have been very much in vogue in recent years. The literature is full of attempts to correlate various metrics of land cover and land use with various metrics of pollinator health. Now, nutrition is almost universally invoked in such studies to explain correlations between landscape and pollinator health. Yet floral resources as such are almost never measured or modeled directly. They hover latent behind land cover classifications with which they are presumed to be correlated. The explicit study of flowers as food, particularly at spatial scales relevant to the ecology of flower-visiting insects, is represented only by a sparse trail of isolated studies. Yet it is almost impossible to overstate the ecological and evolutionary significance of the trophic interface between flowers and insects. When flowering plants rose to prominence in the mid-Cretaceous about 100 million years ago, the relationship between insects and flowers was actually already ancient. The key innovation of flowering plants, angiosperms, was actually not that they bore flowers, for the homologous flowers, sensillato, of gymnosperms not only existed, but were already associated with insect pollinators when angiosperms arose. Rather, it was the edibility. Don Frame makes this argument really compellingly, and especially the edibility of their flowers that distinguished angiosperms from their predecessors and paved the way for their rapid co-diversification with pollinating insects. So angiosperms, more than any plant lineage before them, succeeded in harnessing florivory, which I include the consumption of nectar and pollen as a special case of florivory. Angiosperms harness florivory, this basically bad phenomenon of getting eaten, and they turned it into an adaptive phenomenon by harnessing it toward pollination. And this innovation established one of the chief foundations of global biodiversity. So today, the roughly 369,000 species of flowering plants and 352,000 species of flower-visiting insects together account for a third of all described extant species of eukaryotic life on Earth. So those curves of floral resource availability through time, space, cross-functional traits, those curves hold up the world as we know it. And what we commonly understand as pollination ecology is underpinned by the trophic ecology of flowers and florovores. I explore all these ideas extensively in a synthesis paper that I've just submitted, co-authored with Aaron Iverson and Ingolf Stefan de Winter. At the heart of this paper is a polished-up version of those back-of-the-napkin sketches that emerged from my work in Philadelphia. Floral resource structure can be understood as curves through time, mosaics in space, together distributed across multidimensional floral trait space. All right, now, in case all this seems like merely an abstract theoretical exercise, I want to apply it to a very practical problem. It is a problem that strikes uncomfortably close to home for me, in fact, and one that is acutely relevant to the conservation of pollinators in urban landscapes. Do pollinators compete with one another for limiting floral resources? And in particular, do managed honeybees threaten wild pollinators via resource competition? The topic of competition between pollinators goes back to the 1970s during the heyday of classical niche theory. But back then, the question was purely a matter of academic interest. More recently, however, it has become a topic of public debate and heated polemics. Despite all this attention, though, it has been surprisingly hard to pin down answers. Intuitively, competition should happen, but empirical evidence has remained stubbornly equivocal. My contention is that all this makes perfect sense when the question is approached from a trophic ecological perspective that begins with the multidimensional dynamics of floral resources. Any claim of resource competition is predicated on the assumption of resource limitation. If I ask you abstractly, are resources limiting in, say, Philadelphia, it's hard to get traction on that question, right? But if I ask you the question and show you this graph back here, some uncertainty remains, of course, but you can say with confidence that resources are much more likely to be limiting in July and in August than in May or June. And if we include the dimensions of space, it would be clear that resource limitation is more likely in some places than in others. And finally, if we distributed this temporal and spatial variation across the functional space of the floral community, represented in this diagram by these compartments here, it would be obvious that resources are more likely to be limiting for pollinators that require, say, short corolla depths because they have short tongues, than for longer tongue species that can access deeper flowers. In short, when you begin with the ecology of flowers as food, it is clear that resource limitation and therefore competition is a complex and dynamic ecological contingency rather than a simple yes-no question. But for all its complexity, it is also tractable to the extent that you can estimate the structure of floral resources in the dimensions of time, space, and functional traits. Okay, enough of home office. I promised you we would get back into the field. This portion of the talk will be brief, but I hope it will be compelling. This summer, I set out to explore the question of resource limitation empirically by measuring one of its corollaries, resource depletion. When resources are limiting, the standing crop of floral resources, in this case nectar, should be substantially depleted by insect visitation. To the extent that resources are not depleted, the idea of resource limitation becomes implausible, and with it, the claim of competition. So to do this, I resorted to what is arguably the quintessential method of pollination ecology, namely bagging flowers. So my study sites were located around here. This is central Germany. My University of Würzburg is near the center of this square. And my study sites were a set of protected grassland remnants, most of which could be described as calcareous. So they look something like this. Now, I went out uncomfortably early in the morning and bagged flowers from the most abundant flowering plant species at each site around sunrise, which, it must be said, is really, really early in July in Germany. But the important thing is that this is before appreciable insect foraging had begun. And my sites tended to be dominated by Centaurea scabiosa, but I also sampled Dianthus cartusianorum, Canascia, and scabiosa, which I realized at one point I could not distinguish in the field, so we're pooling them. Echinops sphaerocephalus, Origanum vulgare, and Toicarum comadris. So after bagging the flowers, I returned several hours later in the late morning, and I sampled nectar using microcapillary tubes. Here's an up-close look at the bagging. So these are basically mosquito net bags, and here you can see a bee that has been effectively excluded. I always felt bad for these bees. They wanted that nectar, but it had to be done. I sampled the nectar using microcapillary tubes and measured the volume with a digital caliper. And this process typically took an hour or two and fell between 10:30 a.m. and 12:30 p.m. So this is like a late morning, early afternoon sample, having bagged the flowers in the morning. With these measurements, I compared the nectar volumes found in open versus bagged flowers to estimate the proportional depletion of the standing crop of nectar by flower visiting insects. So, makes sense? All right, now, at this point of the talk, I really wish I were present in person because I would ask you what you would expect the outcome of this study to be. But take a moment and ask yourself, How much of the standing crop of nectar do you think should be depleted in the first several hours of foraging each day? 50%? 75? 95? And why? What is the basis for your guess? Well, did anybody guess almost 100%? Because if you did, you're right. Almost every open flower I sampled across all sites and species was empty, or at least contained so little nectar that I could not recover it with a microcapillary tube. While the bagged flowers, in contrast, consistently held substantial amounts of nectar. The one exception to this rule was dianthus, in which bagged flowers and open flowers held similar volumes of nectar. So I did get appreciable nectar from open dianthus flowers, and I think the reason is that dianthus has these really deep, leathery corollas that effectively exclude bee visitors, and I think even nectar robbers, because they're so tough, and they're visited really only by butterflies. Now, to be frank, I was shocked and not entirely happy with these findings. I still remember my first day in the field when I was sampling what appeared to be a rich flowering meadow humming with bees and butterflies, an ecological feel-good story, right? And then I remember the realization gradually setting in as I sampled one flower after another, that this was actually a scene of desperation. The bees and butterflies were visiting empty flowers. Now, perhaps they were getting lucky one time in 100. Maybe they were merely subsisting on traces of leftover nectar or consuming nectar as the flowers secreted it, leaving no accumulated standing crop. In any case, this was clearly a scenario of extreme floral resource limitation, and one in which the species present must have been in competition with one another. And this is where an interesting twist happens. What were the species present in these grasslands? Well, it was a diverse assemblage dominated by bumblebees and butterflies, but also including various halictids, andrenids, syrphids, even beetles, and the occasional katydid. Almost entirely absent, however, were the prime suspects, honeybees. And this is despite the fact that earlier transect sampling at these sites had found honeybees in relatively high abundance. So colonies existed in the landscape, but by the time I went out to sample nectar in July, honeybees were no longer foraging in the grasslands. And at this time of year, there were no flowering crops or trees providing resources elsewhere. So it appears that without the help of honeybees, bumblebees and butterflies were capable of depleting nearly the full standing crop of nectar each day, and indeed in the first few hours of each day. Here's what I think happened. During this past summer, there was a severe drought across most of Europe, including Germany. In the spring, floral resource conditions were probably still fairly good, but by July, plants were clearly water-stressed, and only drought-resistant species like the ones I sampled were producing nectar. Most flower visitors, like the bumblebees and butterflies that were abundant at my sites, have very little recourse under such conditions. They must forage for the resources that are there, however scant they may be. But honeybees are exceptional. During times of scarcity, they have two options that other pollinators do not. They can stay home and consume stored honey, or they can rob honey from neighboring colonies. And I suspect both of these are better options than visiting empty flowers. This suggests that there might be a sort of release valve to honeybee-induced resource competition. Honeybees will participate in drawing down abundant resources, but they drop out of the competition, as it were, once resources become scarce. Now, of course, this hypothesis requires further investigation, but I think it makes sense of the data that I saw and is consistent with what I know of honeybee foraging biology. All right, I promised to return to Philly before we wrap things up. What have the grasslands taught us about the city? This is speculative, of course, and I now have a more concrete sense of what these colony weight curves mean. So during these periods of abundance that we see in May and in June, floral resources may or may not have been limiting for pollinators in Philly. That's hard to judge. But these periods of scarcity in July and especially in August, and then again at the end of the foraging season, these periods of scarcity probably reflect conditions similar to those that I saw in Germany, because it is during these periods that robbing between honeybee colonies becomes common in Philly. And indeed, in temperate parts of the world in general, if you talk to beekeepers, this is axiomatic. During the summer dearth, robbing begins. Minimally then, in cities like Philadelphia, pollinators can be expected to compete for floral resources during summer dearth periods. Honeybees likely accelerate the onset of these dearth periods by driving the depletion of rich resources. But as competition intensifies, it may be that honeybees become less interested in flowers and more interested in their neighbors. And this adds yet another dimension to the trophic ecology of flowers and florivores. Thank you for your attention. I will be on the Zoom call for the question and answer period if you have any questions. Thank you.